All right, so I would like to start by thanking the organizing committee for picking my research to present, and I'd like to thank all of you for attending this session. My research is weird. It deals in the topic of epigenetics, and the big picture is we're just trying to figure out why does a microRNA become dysregulated in diabetes. So diabetes, I like this definition, it's characterized by the absolute or relative lack of insulin, typically resulting in hyperglycemia. We've gotten pretty good at treating high glucose. However, some patients are still at risk for secondary complications, such as retinopathy, nephropathy, cardiomyopathy, and neuropathy. It's affecting over 300 million people worldwide, that's diabetes, and so finding additional treatment strategies to treat these long-term complications is important. Now, my research focuses on diabetic retinopathy, and it is a microvascular complication resulting from hyperglycemia. Large-scale studies have shown that tight glucose control can actually prevent a lot of the changes associated with diabetic retinopathy. In the early stages, it's characterized by endothelial dysfunction, pericyte loss, permeability changes, and increased matrix production. This results in microaneurysms and damage to the blood vessels in the eye. In late stages, we actually see proliferation of these blood vessels and vision loss. And one of the main pathogenic factors is vascular endothelial growth factor, VEGF. And current therapies that target, or that are for diabetic retinopathy, target this factor. So we're not going to go through all the signaling pathways involved with hyperglycemia. We don't have time for that. However, there are many internal signaling cascades that are affected, as well as transcription factors. Oxidative stress is involved, and this results in increased production of growth factors, such as VEGF and endothelin, inflammatory mediators, such as the interleukins, as well as matrix proteins, such as fibronectin and collagen. These become upregulated. This results in an altered microenvironment, which causes structural and functional changes, leading to vision loss. And in our lab, one way we've looked at trying to regulate these factors is by looking at microRNAs. So what are microRNAs? They are typically 22 nucleotides long, and they are post-transcriptional regulators. They typically target genes for repression, okay? And in the central dogma of DNA, I like to think that they inhibit in between this translation step from RNA to protein. And they can either cause inhibition of translation or full degradation of the RNA molecule. They target they have many targets within the cell. One single microRNA can target hundreds of different mRNA molecules in the cell, so their interactions are extensive. And I like to think they coordinate cellular events. When a microRNA becomes dysregulated, the consequences can be quite extreme. So, in a disease state, we typically see two trends. We see, relative to normal healthy tissue, a microRNA can increase in expression. This causes decreased target gene expression. Remember, they're negative regulators. And this is typically characterized as a gain of inhibition on protective factors. The other trend, and this is the trend we'll be studying today, is that microRNA expression becomes decreased. This results in increased target gene expression and loss of inhibition on pathogenic factors. The green light has gone out. Okay, so one microRNA we've studied in our lab is miR200B. And miR200B is important because it targets VEGF. Remember, VEGF is very important in diabetic retinopathy. VEGF, this is from rat data, and what we see is that VEGF expression is increased in retinal tissue from diabetic rats relative to normal controls, and miR200B is downregulated. And one experiment we've actually done is we've supplemented these microRNAs. We've taken diabetic rats and injected the microRNAs back into the eye, and we've seen that it has protective effects. It decreases permeability as well as corrects the increased VEGF expression. The question for me, though, when I started in this lab is, how is microRNA 200B regulated? Why does it become repressed, and can we target that with some sort of therapy? So, to answer that, we have to talk about another epigenetic mechanism, and that's histone methylation. Histone methylation, some rules before we get into it that are good to know, is that it can be activating or repressing depending on the residue that's methylated. For example, when the fourth residue, which is a lysine, on histone subunit H3 becomes methylated, that opens up the chromatin. It causes gene activation. However, when the 27th residue is methylated, that closes the chromatin, causes gene repression in that area of the genome. It's reversible and dynamic. We have these things called demethylases, and they oppose the actions of the methyltransferases and can remove that methyl mark. Methylation is also thought to be involved in metabolic memory. This is kind of like imprinting. It's the idea that high blood sugar causes changes in these methylation marks which can persist even after blood glucose levels return to normal. What this does is it causes changed gene expression even after the patient has corrected their blood glucose. And finally, only 2 types of methylation have been studied in diabetes, and so it's a relatively new area of study. My research focuses on this Polycomb repressive 2 complex. It is a methyltransferase complex, and the major components are EZH2, EED, and SUS12. It catalyzes this H3K27 trimethylation reaction, and I want you to remember that this is a repressing type of methylation. It's involved in cell survival, adhesion, and angiogenesis, and it's opposed by these demethylases. And so you can see this methyltransferase comes in, methylates the area of the genome, causes closed chromatin, and thus gene repression. These demethylases can remove it. Why study this one in diabetes? In cancer, it's been shown to target miR200B for repression. Cancer is not diabetes. They're very different diseases. However, the underlying biochemical events that are occurring might be the same, and that's why I chose to study this one. Importantly, it can be inhibited with small molecules, such as this one, DesNep. We will see this later in the presentation, as well as ECGC, which is, of course, a compound found in green tea. It also has interactions with long non-coding RNA, and this opens up the future research into new fields. Most importantly, PRC2 and H3K27 methylation remain uninvestigated in diabetic complications, and that's where I wanted to come in. So my hypothesis is that PRC2 expression activity is augmented in diabetes, leading to alterations in miR200B expression, and inhibition of this complex can slow or prevent the development of diabetic retinopathy. Visually, hyperglycemia, I believe it increases PRC2 expression or activity. This increased expression or activity represses 200B through its methylation mark. This will increase VEGF expression. Remember, loss of inhibition increases VEGF expression. And this mediates permeability in the early stages and angiogenesis in the later stages of diabetic retinopathy. So to study this, I've used human retinal microvascular endothelial cells. They are endothelial cells. They line the blood vessels. We've used endothelial cells for many years to study diabetic complications, and this is the first time we're actually using the retinal cell line. My treatments include normal glucose, 5.5 millimolar, what's hopefully going through all of our healthy blood vessels right now. High glucose, which is representative of quite diabetic conditions, 25 millimolar D-glucose, as well as an osmotic control, which uses L-glucose. And this is to tease out whether it's occurring to metabolism of glucose or simply due to osmolarity. My durations are 24 and 48 hours, and that's incubating them with the glucose. I will also I've done some loss of function studies using a chemical inhibitor as well as silencing RNA. I've looked at cDNA from past rat and mouse experiments for correlational evidence, and my readout techniques will be reverse transcription, quantitative PCR, as well as some ELISA and some CHIP qPCR. And if you see a star, that just means the p-value is less than 0.05. So let's get right into it. What we see here, the blue is normal glucose, red is high glucose, and green is the osmolar control. And what we see is that in high glucose, microRNA miR200B becomes downregulated. We see a loss of it about 30-40% in both 24 and 48 hours. And in parallel, we see an increase in VEGF expression. And we would expect this. Loss of inhibition mediates increased VEGF expression. You'll notice that the increase of VEGF at 48 hours is quite modest. However, it is statistically significant. Now, in the same samples, I've looked at the methyltransferase components, EZH2, EED, and SUS12. And this is all mRNA qPCR. And so what we're seeing here is that in high glucose, EZH2, EED, and SUS12 are all increased at both time points. And this provides increased evidence that perhaps this complex is involved in regulating this microRNA. Now, on the flip side, I've looked at the demethylases. And what we can see is that at 24 and at 48 hours, there's almost no change between the demethylases. The methyltransferase components go up, but the demethylases stay the same. Or in fact, it looks like at 48 hours, KDM6A, which opposes this type of methylation, it appears to go down. So what we might expect is a net increase in H3K27 methylation activity. And this is some early data, but it does suggest that is the case. This is an activity assay, and it shows about a 30% increase in H3K27 methyltransferase activity, as well as doing some CHIP qPCR. So what I've done is I've pulled down this methylation mark, and I've done PCR against the promoter region of miR200B. And what we can see is about a 60% increase in methylation at this promoter region in high glucose. The red is high glucose, the blue is normal glucose. At the same time, we see decreased RNA polymerase II at this same region. And this makes sense. If the gene is being repressed, it has increase of the repressing type of methylation, we would expect it to be less accessible to RNA polymerase, less accessible to transcription. That's what we see in the qPCR data, that's what we see with a chip. The biological replicates are low, we don't have error bars, and I've been working to get additional samples, and so we can talk about significance later on. Some loss of function studies, I want you to focus on the last two bars right here. And what we see is that when we give the chemical inhibitor DesNep, we see an increase in miR200B expression in parallel with a decrease in VEGF expression. And this provides some causal data that perhaps this complex, PRC2, is regulating miR200B. When we inhibit it, the microRNA expression goes up, the target of the microRNA goes down. That's nice. However, chemicals are not specific, right? We want to do something more specific. And so using silencing RNA, what I've done is knock down EZH2, the methyltransferase component, and there's no change in 200B. This is sort of an interesting result, not what I expected. However, when I knock down another component, 200B goes up, VEGF goes down. This is very interesting, a new piece of data, and it's something that I'm still teasing out what is going on. Finally, these components are overexpressed in the rat and mouse retina, and so this mechanism might have significance in vivo. So to summarize, EZH2, EED, and SUS12 are increased in retinal endothelial cells in high glucose, as well as the retinal tissue from the diabetic animals. The additional experiments suggest that PRC2's activity is increased globally and at the miR200B promoter region, though we need to confirm this with additional replicates. And finally, the causal data shows that that PRC2 can regulate it, and perhaps through an EZH2 independent mechanism, but we still need to figure that out. So data from this project demonstrates a regulatory relationship between histone methylation and microRNAs, which is a new sort of phenomenon, and understanding such pathways will potentially yield new treatment strategies. I'd like to thank the Chakrabarty Lab, as well as my advisory committee, the Department of Pathology, these funding organizations, and you guys for listening. Thanks a lot.